Hello, AP Bio. In this video, I'm going to introduce the concept of gene regulation. So consider that all of the cells in a multicellular organism all have the exact same genome, but all cells are not the same. We have different cells. We have skin cells and bone cells and cells that protect us in our immune system, and cells that transmit signals in our endocrine system, and then also in our nervous system. In any case, all of those cells have the same genome, but different genes are turned on and off, and we talk about the genes being turned on and off as the control or the regulation of gene expression. And differential gene expression just refers to that concept that all cells that make up multicellular organisms have the same genome, but the different cell types result from different genes being turned on and off. Now, if we think about those different types of cells, let's look here in this slide right here. This is a neuron. In the same organism, we see these types of cells too. Here's a red blood cell, a white blood cell, and a platelet. And you can see how even with the same genome, we get really different cell types. If we look over here in plants, we can see the same thing. On the top, we see the anatomy of a leaf. You can see the different types of cells here. They look really different. Here's the epidermis. That forms one tissue. And then the next tissue is this layer here. These are all the palisade cells, and we have the mesophyll in here, and then the lower epidermis. But you can see how the cell types look really different. These different types of cells, these different tissues come together to form an organ, which has the function of collecting sunlight for the process of photosynthesis. And then here we have a cross-section of a stem, and you can see the different types of cells here too. So it's not just in animals that you see here, but it's also in plants that you see here. You see differential gene expression in multicellular organisms, which brings us to thinking about the purpose of control. It actually depends on whether or not we're talking about unicellular or multicellular organisms. In both unicellular and multicellular organisms, differential gene expression allows for response to the environment. When the environment changes, the behavior of an organism can change in response to that. And those environmental changes can be either internal or external. We talked about external responses to the environment. We have responses to the environment here in the immune system, thinking about response to pathogens of your acquired immune system or even of your innate immune system. But in prokaryotic cells or single-celled organisms, really it's just for response to the environment. But the purpose of control of gene expression, like I mentioned just a moment ago, in multicellular organisms is for cell specialization. So we've got two different purposes depending on whether or not we're talking about multicellular or unicellular organisms. Now, if we think about how cells are organized, just for a minute, or how biological multicellular organisms are organized, the hierarchy of organization of cells, remember that cells, we start with cells, that's the most basic unit of structure and function of all living organisms. And then cells that are all expressing the same genes are going to collect into tissues. That's what you can see here. This is a tissue, the epidermal tissue. This right here is the palisade tissue. So those are all the same cells. They're all expressing the same or similar genes, and they all have a similar function. Collectively, they have a function together, but they all look the same. Now, and we call those tissues. Tissues organize into organs. You can see that really clearly here. This is organized into a leaf. That's the organ, and they have a common goal. Those different tissues have a common goal together that form that organ. Like, for example, in the leaf, we have the purpose is for photosynthesis, and then organs are going to come together in organ systems, and organ systems are involved with solving collective problems, like your digestive system solves the problem of digestion, and your respiratory system is for gas exchange, etc. And then those systems organize into multicellular organisms. So when you're thinking about control of gene expression, think about that. I'm just wanting you to think about that many cells that are grouped together, or not always grouped together, are going to be expressing the same genes because they're the same type of cell, and we call those collections of cells, all the same type of cell, we call those tissues. Okay. What I want to talk about next is a brief comparison between prokaryotic and eukaryotic cells. A little bit of review, a little bit of new layered on top of that. We've talked about this many, many times. Remember, in prokaryotes, let's organize here in this table here, we have 1 to 2 naked chromosomes, meaning that they're not wrapped around histone proteins. They're just chromosomes that are supercoiled. Oh, you learned a little bit about that in chapter 5, and we've talked about that before, that the chromosomes are not contained in a membrane-bound structure like they are in eukaryotic cells. We have multiple chromosomes, so here we only have 1 to 2. Over in the eukaryotes, we have multiple chromosomes, and then those are wrapped up around histone proteins into that nucleosome. So that was the beads on a string. That's that chromatin packing into that 30 nanometer fiber. Now let's add on another layer, another difference between the gene control in prokaryotic cells versus eukaryotic cells. We can look over in this diagram here. In prokaryotic cells, the default position of genes is on, which means that the transcription can begin. If you remember in transcription, or just to remind you that RNA polymerase has to bind in order to start the process of transcription. That's the making of the RNA. And in prokaryotic cells, RNA can bind to the promoter, and then that can slide down and make the messenger RNA. That's the very first step of the gene expression. And in prokaryotic cells, the default is on. Now, the only way to turn it off is if there's binding of what are called repressor proteins to this switch sequence right here that's called an operator. We're going to talk about it later, but just keep in mind that the default position, when there's nothing bound to the DNA, the plain, you can think of it as the plain DNA, is on, and that it gets turned off by the binding of proteins. And if we just contrast that to eukaryotic cells, in eukaryotic cells, the default position is actually off. If you think about the process of transcription, in order to begin for RNA polymerase to bind, we have to have this whole, the DNA bends around. All the regulatory regions are bound with all these transcription factors. This forms the transcription initiation complex. So now the position for the turning on of genes is with the binding of proteins, and that's going to turn the proteins on. RNA polymerase can now bind with the formation of the transcription initiation complex. RNA polymerase can slide down the transcription unit and transcribe the gene, the beginning of the expression of that particular gene. So again, there's a difference. The default position is off, meaning with the naked DNA here with nothing bound. There's the transcription can't begin in eukaryotic cells, but it's opposite in prokaryotic cells. When the DNA is quote unquote naked, it's on, and then the protein binds and that turns it off. So it's sort of opposite, or totally opposite, not sort of opposite. Okay, other differences is thinking about transcription and translation in prokaryotes. Remember that there's no nuclear envelope, so as soon as the messenger RNA is transcribed, then the ribosomes can bind to the lengthening, the elongating messenger RNA. We say that transcription and translation are coupled because they can happen at the same time. Transcription has to obviously start slightly before, but they are happening at the same time. As opposed to in eukaryotes, transcription happens inside the nucleus. DNA is transcribed into RNA, and then RNA then can leave, and a ribosome complex will form, and then we get the translation of the protein in the cytoplasm. So whether or not it's a bound ribosome or a free ribosome, all happens in the cytoplasm. They're bound to the rough endoplasmic reticulum or free floating in the cytoplasm. So that we say that transcription and translation are sequential. So there's another difference. A fourth difference we see here, remember in prokaryotic cells, there's no RNA editing, also called RNA splicing, also called RNA modification. Those are all the same thing. We have the messenger RNA comes off after being transcribed from the gene, and it's already ready to go. It's already mature. However, in the eukaryotes, we have RNA editing, RNA splicing, RNA modification. Those mean all the same thing. The ends are protected. The 5' cap is added. which is just a modified guanine. The poly A tail is added to the end, so that's protecting the ends of the messenger RNA. And then the introns are cut out and the exons are joined to make a mature messenger RNA. Then after RNA is edited, it can leave the nucleus and then it can be transcribed on the ribosome. So no RNA editing over in the prokaryotes, but we have significant RNA editing over in the eukaryotes. Now, because of the complexity, the difference in complexity of prokaryotes versus eukaryotes, we have different places where the process can be controlled. Again, recall that in chapter 18, you're reading and learning about the control of gene expression, the turning of genes on and off. And because it's simpler here, where we have coupled, we have the default being on, and we have no RNA editing, there's much less DNA. We only really have three main points of control in the prokaryotes. And we're going to talk about that in a little bit in another video. But over in the eukaryotes, because this is a more complex process, we have multiple points of control. So this can be turned on and off at multiple places along the process of gene expression. Now, even though there are multiple places of control in both of these, and we have more complexity over here in the eukaryotes, the most common point of control is still transcription. So if you don't need milk, don't stop at Lampman's on the way home. In other words, messenger RNA is not even made if the gene isn't turned on. So that's the most common place of what we call gene silencing, turning a gene off or turning a gene on. We call that regulated transcription because it's regulating at the point of transcription, the messenger RNA doesn't get made. Now, that doesn't mean that all transcription is regulated, and that certainly doesn't mean that all genes are regulated at transcription. It's just the majority for both prokaryotes and eukaryotes. The type of transcription that's unregulated, we call that constitutive transcription, and that just means genes that are just expressed all the time. They're not turned off in prokaryotes or eukaryotes. And it depends. Different genes are turned on all the time in different organisms. In eukaryotes, some of the genes that are turned on all the time are, for example, the proteins that are involved in the process of glycolysis. That's happening all the time. All eukaryotic cells are running on ATP. Glycolysis has to happen in all cells. Those are genes that are constitutively transcribed on all the time, and that's not regulated transcription. Now, thinking about how transcription is regulated in prokaryotes and eukaryotes, that is actually different. So even though the most common point of control is the same, the mechanism for control is different. In prokaryotic cells, and only in prokaryotic cells, we have something called the operon. And the operon refers to a cluster of functionally related genes, meaning they're located next to each other on the DNA, located one after the other. And you can see here's a cluster of functionally related genes. Here we have a cluster of functionally related genes. Here we have a cluster of functionally related genes. So they're all have some type of related function. That's where they're called functionally related. They're grouped sequentially into a single transcription unit. So they're all right next to each other in the genome, which makes on the chromosome, which actually makes a lot of sense. In eukaryotes, it's actually not the same. The genes are just everywhere in the genome. They're scattered throughout. And so there isn't any mechanism like the operon. Oh, by the way, sorry, in the operon, these are controlled by a single on-off switch. We call that on-off switch an operator. And we're going to go into detail in that in the next video. But just know that the operon is a mechanism of transcriptional control, regulated transcription in prokaryotes only. But if we go over here to eukaryotes, again, sorry, the eukaryotes don't really have that. So the genes are spread out on different chromosomes, even if they're related to the same process. Say, for example, you're trying to produce insulin. The insulin gene is not near the other enzymes that are responsible for the process. Everything is everywhere, but they're grouped together into what are called transcription factories. So areas that are really far apart in the genome on totally different chromosomes, they can be brought together in the nucleus and then grouped together with RNA polymerase so that they can all be transcribed and turned on at the same time. So there are these little organized, I think of them as little tiny organized kind of like little hairballs within the genome of different pieces and parts of the genome that are pulled together. And we call those transcription factories. So they're a cluster of functionally related genes located on different chromosomes with RNA polymerase together, and then also the same transcription factors and all those other things. Okay, that's it for now for an introduction on the regulation of gene expression, and I'll introduce the next concept.